Okay, so we like to discuss everything as chromosomes because chromosomes is the collection of genes. Okay, and that's basically what, a singular circular chromosome is basically what we're going to see in most prokaryotes, bacteria and archaea. Are there going to be some exceptions? Sure. Can't think of any off the top of my head because there's so very few, but can't sit there and say no. That can't happen. Now, eukaryotes, for all intents and purposes, going to have more than one chromosome because most part we're diploid, triploid. Yes, I know some plants are haploid, but they're going to be diploid during parts of their life cycle. And our chromosomes tend to be linear. Again, I say tend to be because yes, there's going to be some eukaryotes that have circular chromosomes. Okay, can't think of anything, but I'm not going to sit there and say no. So when you look at the genomes and you compare the genomes, as we talked about, chapter 5, viruses. Viruses just basically break all the rules. Okay, single, double-stranded DNA or RNA, relatively short, measured in thousands of base pairs. Our prokaryotes, double-stranded DNA, longer, relatively longer than the viruses, generally circular, measured in the millions. Us eukaryotes, double-stranded DNA, extremely long, pretty much linear, extremely long, measured in the billions, B, for the most part. Mitochondria, chloroplasts, which also carry their own genome. Okay, remember that. The genes for how to do stuff in the mitochondria, how to make the proteins, how to make and maintain a mitochondria, and how to make and maintain a chloroplast are carried within them. The mitochondrial genes are not found in your nucleus. Remember, that's part of the whole, what is it, proof that we have of the endosymbiont theory. It's a supporting information. Pretty much all organisms are going to have some form of a plasmid, which we'll be talking about in this chapter. This is an extremely small, circular chunk of DNA, measured in the hundreds, tops out at a few thousand at best. All organisms on this planet that we know of have transposable elements. This is now, when we look at this double-stranded DNA, it's going to be maybe the size of partial gene, an intron, the intervening sequences. A lot of those were, these transposable elements 100 million years ago that just got stuck because they can pop in and pop out. But hey, we'll talk more on those later. So these plasmids, circular, linear, double-stranded DNA. Why the circular or linear? Again, depends on which archaea, depends on the few bacteria it may be there. Most part, when you see a linear plasmid, that's going to be in the few that show up in eukaryotes. As I said, these transposable elements are going to be segments of DNA that gets inserted into a chromosome. It's a small chunk. At best, it's the length of a gene. At worst, it's only a fragment of a gene. Gets inserted. Inserts, pops out. How often does it insert, pop out? Well, it depends on what the transposable element is, what the host is. There's theories now that pretty much the majority of those introns, those intervening sequences in our human chromosomes, are remnants of these transposable elements, remnants of viruses from hundreds of millions of years ago, and we've just accumulated them over millions and millions and millions of generations. We study most of this in bacteria because bacteria are so easy to grow. They're so easy to alter genetically. Because think about it. To do this, do a lot of genetic experiments on eukaryotes. You have to take into account. Okay, if it's animal, it means it's going to be diploid for the most part. A few insects may be haploid, but we're not going to worry about those. And then you've got genes spread across numerous chromosomes. Diploid, multiple chromosomes, genes spread around. Ugh. Whereas bacteria, remember, measured in millions. Yes, there are still quite a few genes, but nowhere near ours. 4,300, they think, in E. coli, making up about 88% of its genome. The remaining 12%, that's going to be expression elements, promoters, different, binding sites, regulatory elements. We see that the genes in bacteria, though, do something that doesn't happen in us eukaryotes, especially us mammals, as animals. These genes are going to be clustered together in what are referred to as an operon. If there's a set of genes that need to be expressed at the same time due to the same stimulus, they're going to be collected physically together in this operon. And for the most part, that operon, all the genes, the 2, 3, 4, 6 genes, whatever, are all going to be transcribed on one messenger RNA. Ruh-roh. That's so much different than us. For us, every pre-mRNA is only one gene. After you do the splicing and all that to get rid of the introns and splice all the exons together, you have a mature messenger RNA that is just one gene. Well, with prokaryotes, since they don't have the introns, exons, and they're going to have a lot of their genes grouped together in these operon things, one long messenger RNA that they call polycistronic. Polycistronic, meaning it has multiple genes on one messenger RNA that they will all be translated one after another. Hell, some of them could be translated as one really long polypeptide that gets cut into individual polypeptides that will then fold into the mature protein. So when you look at E. coli, there may be some differences between the E. colis. Notice here it says strain K12, genus, species, strains. The genetic sequence, the AGT sequence, may be different between the strains. Well, will be different between the strains. But generally, where the genes are on this chromosome will relatively be conserved. Notice the 2, 4, 6, 8, 10, 12. This is hundreds of base pairs, 200 base pairs, 400, 600, 2400. So that's 2.4 megabytes. 42, 4400 megabytes. So 4.4 megabytes. 4,400 kilobytes. So that's 4.4 megabytes. Mentioned in that few slides ago in that table, this idea of plasmids. Plasmids are going to be found throughout the prokaryotes. Plasmids are easily shared between bacteria. And we've just found out over the last couple of decades, we've known about plasmids for quite a while. I think since the 50s, 60s. But within the past couple of decades, they've been studying them more and been able to use better molecular tools in the lab to study how they're passed. And when I was an undergrad, back in the mid-90s, It was taught to us, if I remember correctly, that plasmids get passed back and forth, but it's only between bacteria of the same species. Now, I'm going to be talking to you about this and telling you that the bacteria can pass it between species. E. coli to Salmonella, Salmonella to Shigella, Rutro. That could cause some problems. There's also, though, what causes problems as we're trying to study it, it also explains how some of this antibiotic resistance has been able to spread so easily and so quickly through different species. It's popping up in different species. Now, when these chromosomes, I'm sorry, not the chromosomes, these plasmids, I mean, about 5% the size of a normal chromosome. They're not essential, okay? They're not carrying any specific gene that is needed metabolically. Oh my God, here's the gene for, the making of, isocitrate that's needed for the citric acid cycle. No, nothing like that. It's going to be carrying different genes that will enhance the ability of this bacteria to survive and enhance its ability to outcompete other species in its environment. Copy numbers, where generally it's one chromosome per bacterial cell, unless it's on its, in metabolically really active during, that exponential or that log phase growth, and it's getting ready to go through binary fission, then it may have two. But on normal conditions, it's only one. Well, with plasmids, it could be one, it could be hundreds. Depends on what the plasmid is, depends on which bacterial cell is housing it, how many copies will be made. You see from this picture here, here is the chromosome. Boom. Right there, where the arrow is, that little bit right there, those are the plasmids. Here, yes, because of its isolation, the supercoiling of the circular chromosome has been relaxed a bit, but you can see how it folds in on itself. It's, coiled in and around. Plasmids are going to fall into a couple of groups. One group is what I refer to as R plasmids. These are the R comes from resistance plasmids. They confer resistance to antibiotics. Yes, I know, antibiotics. In our hubris, we seem to think antibiotics are our thing. No. The original antibiotics all came from bacteria, archaea, soil microbes, soil prokaryotes. They have been competing together, against each other, I should say, for, a couple billion years. Well, during their competition, they've evolved to have weapons. If they've got weapons, which we call antibiotics, they have to have defenses, which are going to be resistance genes, genes that allow them to nullify antibiotics. Well, turns out some of those genes somehow got off chromosomes and got onto plasmids. Plasmids which could then be passed around. We've now seen that over the billion years of evolution, selection, resistance, has made it so that instead of our plasmid have only one resistance gene, many of them that we've isolated from nature can have multiple resistance genes on them. So they're very, very small. Tet, right there, boom. Right there, that is responsible for tetracycline resistance. It's going to have its own genes responsible for replication and the maintaining of this. What is it? CAT, chloramphenicol. You know, oh shoot, I can't read my own notes. They're so small here. But, each of these different things is a different resistance gene. We also see that some of the plasmids are plasmids, but they're passed around because they give, new abilities. It's a plasmid that's carrying genes that normally wouldn't show up in that bacteria. Certain virulence factors, the ability to make certain toxins. E. coli, 0157H7, the one that causes the bloody diarrhea. Jack in the Box back a couple of decades ago had a couple of little kids who died from eating their burgers because they were contaminated with E. coli 0157H7. You hear nowadays when people get sick, and I'm not talking just diarrhea, I'm talking you are literally shedding the lining of your gastrointestinal tract, your large intestine, due to Shiga toxin. It's the toxin from Shigella dysentery. Somehow the gene for making that Shiga toxin got on a plasmid. That plasmid somehow got passed on to E. coli. Now we have E. coli 0157H7. Well, that's a virulence factor that carried and passed around on plasmids. Not all bacteria can take up the plasmid, and if they can take it up, they can't produce and then, export the toxins. You'll see some have bacteriocins. These will allow for that strain of bacteria that has this to inhibit or kill other species of bacteria in their environment. The virulence factors, like the toxins, those are generally going to be against us. These bacteriocins, those are going to be against other bacteria. And you can see things, certain metabolic abilities, hydrocarbon degradation, the genes responsible for that carried on a plasmid.